Hello students, welcome to my channel Bioscience Guide. Today the topic is embryology or developmental biology of frog. Embryology is the study of development of an organism with the changes through the zygote or fertilized egg. Embryology or developmental biology consists the following steps: prenatal or pre-embryonic development and postnatal or post-embryonic development. Prenatal or pre-embryonic development, which takes place of an organism from zygote up to birth, and it includes the various steps like cleavage, morula, blastula, gastrula, etc. Postnatal or post-embryonic development is the development of an organism from birth up to death. Gametogenesis is the process of formation of gametes. It is also known as the formation and development of either male or female gametes in the male reproductive organ or female reproductive organ respectively. Gametogenesis consists following types: spermatogenesis and oogenesis. Spermatogenesis is the formation and development of male gamete or sperm in the male reproductive organ or testes. Oogenesis is the formation and development of female gamete or ova in the female reproductive organ or ovary. Spermatogenesis takes place in the testes. Each testis is composed of numerous seminiferous tubules. Each seminiferous tubule is lined by germinal epithelial cells or germ cells. The germ cells can modify into spermatogonia. Spermatogenesis, which occurs in three phases: multiplication phase, growth phase, maturation phase. In multiplication phase, the germinal epithelial tissue of testes consists cell which acts as a primary germ cell. Now, the primary germ cells divide repeatedly by mitosis cell division to form primary and then secondary spermatogonia in diploid form. In growth phase, the secondary spermatogonia increase in size due to accumulation of nutrients, and they are now matured in form called primary spermatocytes. In reduction or maturation phase, the primary spermatocyte divides by meiosis first or reduction division to form secondary haploid spermatocyte. The secondary spermatocyte divide by meiosis second to form haploid spermatids. Each spermatid develops as a motile, elongated, thin body sperm. It consists head, middle piece, and tail. The process of spermatogenesis is described as diagrammatically, in which the germinal epithelial tissue of the testes consists cells which act as a primary germ cell. They are diploid in form. Now, the diploid primary germ cell divides mitosis cell division. It forms 4 in number of diploid cells. They are called secondary spermatogonia. The diploid secondary spermatogonia accumulates nutrient and becomes enlarges in size called primary spermatocyte. The primary spermatocyte goes on division by meiosis first or reduction division to form haploid daughter cell. They are called secondary spermatocytes. Now, the secondary spermatocyte divides meiosis second to form haploid spermatocytes. In this way, a primary spermatocyte on meiosis first and second to give there are 4 in number of haploid spermatids. Each spermatid develop as a motile elongated sperm. Each sperm consists head, middle piece and tail. Thank you for watching.